The isolation of functional genes in nucleotide sequence space, evidence for intelligent design. One of the most intriguing aspects of molecular biology is the genetic code, which is made up of nucleotides, the building blocks of DNA. Genes, which are specific sequences of nucleotides, are responsible for encoding the information necessary for the creation of proteins and the regulation of biological processes. But the question arises: how likely is it that functional genes could have emerged through random processes in the vast landscape of nucleotide sequence space? The concept of the isolation of functional genes within this sequence space reveals a significant challenge to Darwinian evolution. Given the sheer number of possible combinations of nucleotides, functional genes are extremely rare in this vast space. This isolation suggests that the random mutation and natural selection mechanisms proposed by Darwinian evolution are insufficient to explain the origin of genes. Instead, this points to the necessity of intelligent design, where a designer purposefully arranged these sequences to create the complexity we observe in life. Here, we will explore the concept of nucleotide sequence space, explain the isolation of functional genes, and argue that this isolation provides compelling evidence for design rather than random evolutionary processes. 1. Understanding nucleotide sequence space. To understand the isolation of functional genes, we first need to explore the idea of nucleotide sequence space. DNA is composed of 4 nucleotides: adenine, A, thymine, T, cytosine, C, and guanine, G, which are arranged in various sequences to encode genetic information. A gene is a specific sequence of nucleotides that provides instructions for producing a protein or for regulating certain cellular processes. Just as amino acid sequence space represents the total number of possible sequences of amino acids in a protein, nucleotide sequence space refers to the total number of possible arrangements of nucleotides in a DNA sequence. For a gene of 100 nucleotides, for example, the total number of possible sequences is 4 to the power of 100, or approximately 1.6 times 10 to the 60. This is an incredibly large number, which illustrates how vast sequence space truly is. However, not all possible nucleotide sequences result in functional genes. The vast majority of sequences are non-functional, either because they do not produce proteins or because the proteins they do produce are non-functional or even harmful. Only a tiny fraction of sequences in nucleotide sequence space correspond to functional genes that are capable of encoding useful biological information. This creates a problem for Darwinian evolution, which relies on random mutations to produce new genetic information. If functional genes are so rare in sequence space, how can random mutations ever find them? The isolation of functional genes highlights the improbability of these sequences arising by chance. 2. The isolation of functional genes in sequence space. The concept of isolation refers to how functional genes are separated by vast expanses of non-functional sequences within nucleotide sequence space. In other words, functional genes are like islands in a sea of non-functional sequences. These islands are extremely rare, meaning that finding one functional gene through random mutations is already highly improbable. Moreover, the idea that mutations could gradually transform one functional gene into another, more complex one, is even less likely, as intermediate sequences are often non-functional. This isolation is critical because it shows that natural selection, the primary mechanism of Darwinian evolution, cannot easily guide the search through nucleotide sequence space. Natural selection can only act on existing functional genes; it cannot preserve non-functional intermediate sequences. If a mutation leads to a non-functional gene, that organism is less likely to survive and reproduce, meaning the non-functional mutation will be eliminated by natural selection. As a result, there is no pathway for evolution to bridge the gap between different functional genes. 3. The improbability of finding functional genes by chance. The vastness of nucleotide sequence space and the rarity of functional genes within it make the random emergence of genes highly improbable. Even though random mutations can theoretically explore new sequences, the probability of stumbling upon a functional gene is so low that it becomes an insurmountable defeater for naturalistic explanations. Consider the analogy of a library containing all possible combinations of letters, where only a tiny fraction of these combinations form coherent sentences. The task of finding a meaningful sentence by flipping through pages at random is incredibly difficult, given the overwhelming number of nonsensical combinations. Similarly, in nucleotide sequence space, functional genes are vastly outnumbered by non-functional sequences, making it improbable for random mutations to stumble upon the precise arrangements needed for biological function. The waiting time problem compounds this challenge. The time required for random mutations to generate a functional gene, even in a single-celled organism, far exceeds the available time since the origin of life. This is particularly problematic for the Darwinian model, which assumes that functional genes gradually evolve through random changes over billions of years. The data suggest that there simply hasn't been enough time for such a process to occur. 4. The failure of evolutionary mechanisms. Several key features of nucleotide sequence space expose the limitations of evolutionary mechanisms such as random mutation and natural selection. Lack of gradual pathways. For Darwinian evolution to work, there must be a gradual pathway from one functional gene to another, where each intermediate step provides a selective advantage. However, the isolation of functional genes in sequence space shows that there are no such gradual pathways. The vast majority of intermediate sequences are non-functional, meaning that natural selection cannot preserve them. Without a stepwise process to guide the evolution of new genes, random mutations would have to generate functional genes de novo, a process so improbable that it defies realistic expectations. Functional complexity requires specificity. Genes encode proteins, which are essential for carrying out nearly all biological functions, from enzyme activity to cellular structure. The functionality of proteins depends on the specific sequence of amino acids, which in turn is dictated by the nucleotide sequence of the gene. This specificity means that even small changes to the nucleotide sequence can render a gene non-functional. The more specific a gene's function, the more precise its sequence must be, and the more isolated it becomes in nucleotide sequence space. This level of fine-tuning is impossible to create through random mutation, as most changes to a gene will likely degrade its function rather than improve it. 5. Intelligent design as the best explanation. The isolation of functional genes in nucleotide sequence space, combined with the improbability of finding these sequences through random mutation, points to intelligent design as the best explanation for the origin of genes. The fine-tuning and specificity required for functional genes suggest that they were not the product of chance, but were instead purposefully arranged by a designer. Just as a coherent sentence in a book requires an author to arrange letters in the correct order, functional genes require a mind to arrange nucleotides in the correct sequence. The fact that functional genes are so rare in sequence space indicates that they were not stumbled upon through random processes but were designed with intent and foresight. From a Christian perspective, this designer is God, who created life with wisdom and purpose. The Bible affirms that God is the creator of all things, as seen in Genesis chapter 1 verse 1. In the beginning, God created the heavens and the earth. The complexity of the genetic code and the isolation of functional genes reflect the handiwork of a creator who designed life to function in harmony with his creation. Conclusion. The isolation of functional genes points to design. In conclusion, the isolation of functional genes in nucleotide sequence space presents a significant challenge to Darwinian evolution. The vastness of sequence space, combined with the extreme rarity of functional genes, makes it highly improbable that random mutations and natural selection could have generated the genetic information required for life. The absence of gradual pathways between functional genes further undermines the Darwinian model, as evolution has no mechanism for bridging the gaps between isolated gene sequences. The evidence strongly supports the concept of intelligent design. Functional genes, with their specificity and fine-tuning, point to a designer who purposefully arranged these sequences to create life. As we continue to study the intricacies of the genetic code, it becomes increasingly clear that life is not the product of random chance but the result of divine creation. Thanks for watching. Please like, share and subscribe, and comment below if you agree or would like to add your thoughts. If you would like me to respond to your comment, please make a level 1 comment directly below the video. I will respond to all polite level 1 comments even if it takes me a while, due to other commitments.